Hello children. In today's session, we will be discussing about the topic called as mutation. But before we go and understand what are the mutations in a little detailed manner and how they happen, let us revise a little bit about a topic which we have already gone through. It is DNA replication. Now, DNA replication, as we all know, is doubling of the genetic material of the living organism. For example, like DNA is a double-stranded structure. So, for the replication of the DNA, this double-stranded structure of the DNA first has to unwind itself. Then both the strands they separate out, and both the strands they replicate or they make their own copies. So once they make their own copies, we all know that the one strand remains in the new genetic material. The first strand, one strand will be the mother DNA which was originally present, and another strand will be the newly synthesized DNA, newly synthesized strand. How the synthesis happens? That on the mother DNA, whichever nucleotides are there, like adenine, guanine, cytosine and thymine, the complementary base pairs of those nitrogenous bases are added to the new strand. This is how that replication in general we can talk about happens in the DNA. Basically, when we talk about the replication of the DNA, it is a very accurate process. DNA does not, DNA replication does not make any kind of mistake. By default, any mistake occurs also. We have a proper machinery to rectify it also. That means after the replication of DNA has been done, there are systems which will go through, that is going to inspect the replicated DNA, find out if any discrepancy has occurred, and it will rectify it. It will cut that particular portion and join the remaining DNA. So even after the replication, we have a repair mechanism for the replication. So chances of having any mistake in DNA replication is very rare, or you can say that it is nil. But still our DNA replication can make some kind of mistakes, or you can say that some amount of inheritable change might occur in the DNA replication, and that is called mutation. So what is mutation? Mutation is sudden change in the genetic material, that is DNA. Now this mistake can be of many types. Out of that, 4 types we are studying just now. One, you can see in the slide, one is point mutation, second is frame shift mutation, third is insertion, and fourth is deletion. So before we go into the reading of the definitions of all of them, let us understand this thing one by one. Now the slide which is projected in front of you gives a little idea of all the 4 types of mutation which we have thought about it. First one, you can see the first strand where I am just reading TAA, CTG, CAG and GT. Now what I'm trying to tell you is that just the previous topic which we have did, we have talked about the codons. And when we talk about the codons are always read in triplet forms. So ATT is one codon and which is responsible for the formation of one amino acid. Then next to it, CTG is one codon and it will be responsible for the synthesis of one amino acids. So like that, the whole strand of the DNA, the first diagram you can have a look at it, will be read in triplet forms. This is an ideal DNA, a correct DNA. Now, in the second diagram, just have a look at it. By any chance, if one nucleotide is replaced by another one nitrogenous base is replaced by another nitrogenous base. Look at it. TAA, that remains okay, there is no problem with it. But instead of CTG, that will be the second codon in the original strand. Instead of that, in the second diagram you see that T is replaced by C. So what has happened? The T has been substituted by C. Now in this case what will happen? CTG might be going for synthesizing of one type of amino acid, but once the T is replaced by C, CCG will be synthesizing another amino acid which was not synthesized by CTG. So in this case what is happening, that once the amino acids are being coded, because of the substitution of one nucleotide base pair, another amino acid is coded. So there will be no change in the whole big framework of reading. Just one amino acid gets changed. Original one, just imagine lysine was going to be secreted because of the triplet. Now because of the substitution, lysine is not going to be secreted. Valine is going to be coded. So substitution mutation does not change the whole sequence of the amino acid. It just changes the coding of one amino acids. Now come down to the third one. Third one is base addition, or you can call it as insertion. Now insertion can be of two types. If insertion happen in a codon form, if three base pairs are attached in between. Now what will happen? Once these three base pairs are attached, you can imagine one more amino acid added to a polypeptide chain. But if instead of triplets, only one base pair is added like it is shown in the diagram. See in this case, TAA, that remains same from the original one. Come down to the second triplet which we are discussing. In the original one it is CTG, but in base addition what has happened? Between C and T, one more C is added. So now the triplet reading will be CCT. Then this one more C is added between C and T, so CCT will be read. Then G will be shifted to next triplet codon, so GCA will be read. Then A will be shifted to next codon, so it will be read as AGT, and this T will remain separate out. Now another codon is going to add to it. So now what is happening? Because of addition of one C in between C and T, the whole reading frame of the strand changes. So there will be not change in one amino acid synthesis. Here, because of the insertion of one base pair, there will be change in the whole reading sequence of the strand of the DNA. That means the amino acid which was going to be synthesized, which was going to be coded from the point CTG will be changed, and throughout the whole of the strand it will be changed. So this is called as frame shift mutation, where the reading frame gets shifted, and this is happening because of a type of a mutation which is called as insertion mutation or base addition. Same thing will be happening if you delete a base pair. If you delete three base pair in one go, only one thing will be there, that particular amino acid which was going to be coded by those three base pairs, triplet, will not be coded if it is in triplet form. But if you delete, or because of the mutation, if one base pair is deleted. Now look at the diagram, in this case the fourth diagram. TAA remains constant from the original, it is same. Now what you have to think is that in the original it was CTG, that was the second triplet codon, but in the fourth diagram you can see T has been removed out. So now how the codon will be read? Instead of being read as CTG, it will be read as CGC. Then another one AGG. So what is happening? Once the one base pair is removed, the whole of the reading sequence, the whole of the reading frame of the strand gets disturbed. And it will not affect only the coding of one amino acid, it will affect the coding of all the amino acids. And the amino acids which were going to be synthesized will not be synthesized, because the codons which will be synthesizing the amino acids are totally disturbed when we talk about the deletion or insertion of one base pair. So this will lead to a kind of a process called as frame shift mutation. So point mutation means when there is a change in one base pair. Point mutation can be two types, either insertion or deletion. One base pair inserted, so insertion mutation. One base pair deleted, then it is called as deletion. And in both the cases, what is happening? The whole reading frame of the codons gets disturbed out. So this is actually with the codons I have tried to explain it to you. Let us understand this frame shift mutation along with some English language sentences. I'm just projecting that slide. I'm just moving the slide, and then once we have understood, then only we will go through the reading part of the slides. See, now here you can see the first sentence, the cat ate the rat. Now imagine these are triplets. These are codons which have three base pairs. The means THE, CAT, ATE, THE, RAT. So triplets codons you can see in this. This is the original sentence. First one, what we are going to see is just the substitution. So instead of C, the cat, C, instead of C, K is replaced there. So the sentence will be, the cat ate the rat. So though C is been replaced by K, there is no change in the meaning of the sentence. Because once you pronounce it out, the pronunciation gets the same thing. Come down to second one. The cat ate the rat. So C is now replaced by H. So how it's going to pronounce that sentence? The hat ate the rat. Now in this case, there is a major change in the reading of the sentence. So this from here, the change starts. Next one. The cat ate the rat. This is the original sentence. Now in this case, what they have done, they have removed T of the cat. The second word, that is CAT, T has been removed. So how this whole sentence got shifted? The T has been removed. So A of the 8 will come with CAA. The cat ate her at. So in this case, what is happening? The whole of the reading frame of the codons have been changed. This is because of the deletion of one single base pair. In this case, it is T. Removing of the T, so it has to be triplet only. So THE is there, but CAT removed. So CAA came from the at word. Then once A goes, T came from the word. Then comes the insertion. Insertion, the cat ate the rat. That is the original sentence. Now in this case, what we have done, what has happened? After the, E has been inserted. So ECA, T got shifted to at. So TAT. Then E got shifted to the. So ETH. Then E of the got shifted to rat. So ERA and then T. So in this case, what's happening? Because of insertion of one E in between cat, the cat, there is change in the reading framework. So this is the frame shift mutation, where the whole reading framework for the amino acids gets shifted either because of addition or either because of the deletion of any of the nitrogenous base. Now let me just read the definitions which are there. To start with, what is point mutation? Mutation in a single base pair which is replaced by another base pair. For example, in case of sickle cell anemia, we have already studied in one of our lectures. Point mutation in beta globin chain results in change of glutamate to valine. Insertion, it is addition of one or more nucleotides in the DNA segment. Insertion of 3 or its multiple does not change the reading frame, but only a new amino acid is added. But if it is 1, it will change the whole frame of the reading. Deletion, The removal of one or more nucleotide from the DNA segment, deletion of 3 or its multiple base do not change the reading frame but remove one or more amino acids. Third one, frame shift mutation. It is the change in the reading frame because of the insertion or deletion of the base pair, and that we have understood through this particular slide which is there. Now what we have gone through for this whole explanation, let us revise it out with this one video which I am just putting it after this. Let's have a look at it.
DNA rarely makes mistakes, but it can happen. A point mutation changes a single base. This can change the whole amino acid, or depending on its position, could go unnoticed. Other mutations can be more damaging. A deletion removes a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the deletion, the amino acid sequence will change because the bases shift by one. The resulting protein will likely be non-functional. An insertion adds a base, but also causes a frame shift, which changes every amino acid after the mutation. Originally, the codons would be formed in this pattern. But after the insertion, the amino acid sequence will change because the bases shift by one. Again, the resulting protein will likely be non-functional. Lastly, an inversion will invert a segment of DNA end to end. Watch this section closely. Each base has been moved around. Again, these mutations have the potential to be harmful, but not all mutations have an effect on protein. Remember, during translation, a start codon must first be read to begin coding the protein. If the mutation happens before or after the protein coding sequence, there will be no effect on the protein. Mutations must occur in the coding section of DNA to affect protein synthesis. Otherwise, nothing happens. Like and subscribe, and follow me on Twitter and